right, so like we just talked about, Karen, what do you think these black dots are? If this is my phospholipid, this is the cartoon drawing here, and this is the molecular drawing. What do you think those black dots are? So this is the cartoon drawing on my phospholipid. And so this is the tail, the gray. You see the gray tails? And this is the same drawing as this, only it's the molecular structure. I'm showing you with the dots. They represent atoms, elements on the periodic table. So my question is, what do you think these black circles represent? You want some choices to choose from? All right, so your choices are the black circles hydrogen, oxygen, carbon, or potassium? Okay, you're really close. The blue dots are the hydrogens. The red dots are the oxygens. The carbons. It's the carbon. All right. Not carbon dioxide, but carbon. Remember, this tail is hydrocarbons. So then hydrogens and carbons. The black dots are the carbons, and all these blue dots are on the outside. Those are the hydrogens. Is this tail hydrophobic or hydrophilic?
Say, look it over.
Terry. Hydrophobic. Hydrophobic is correct. So, Kelly, remember the picture? Were the tails on the outside or facing the inside?
They're on the inside.
Which means they got repelled by the water, remember? So because of that, they're hydrophobic. And so here is the slide. Everything we just said, phosphate heads, hydrophilic meaning what, Zach?
Hydrophilic means that it bonds with water.
Right, it reacts nicely with water. And then our fatty acid tails, which are mainly made of what two atoms, Josh?
Carbon and hydrogen.
Carbon and hydrogen, good. And then when they're put in something that's polar, like water, it forms a bilayer. There's the bilayer right there. Did you have a question?
Me?
I don't know if I saw your hand up or not.
Oh, it's because I thought you were going to ask a question and I thought I was going to know
Oh,
the
you're
answer.
gonna have the answer. What were you gonna say?
No, I just thought you were going to ask what the fatty acids. Never mind. I don't know.
Gotcha. I'm with you on that. Does anybody remember the functional group for a fatty acid? Nick?
Lipids. It's hairy.
Harry, sorry. No, that's Harry. You're Nick.
No.
They've started switching names.
No, yeah.
Oh, okay. What is the, okay, so the lipid is the polymer, and the fatty acid is the monomer, and on that fatty acid, on the end of it, there's a functional group. What's the functional group on the end of that fatty acid? Carboxyl group, good. Raise your hand if you remembered that. Yeah, high five everybody.
Good job.
Okay. So the lipid bilayer is one of those structure meets function things. We'll talk about that throughout the entire cell unit when we discuss more in depth the mitochondria and the chloroplasts and the ribosomes.
I love that little
Same.
thing.
Good.
And like when I'm
Cute.
going through the things I'm studying, I'm like,
So Paula, explain to me what you're looking at here.
that's the lipid bilayer.
That's a lipid bilayer. And what is this part up here?
That's the phosphate.
Phosphate heads, right?
Yeah.
And what is significant about the phosphate heads?
They are hydrophilic.
Good. Everybody okay with this picture? If I show this to you on a test?
What is the yellow dot?
Yeah.
Oh, okay, the yellow dots. Who has a guess? Yeah.
Potassium.
Not potassium. That was the third one. Yeah.
The oxygen.
Not the oxygen.
Phosphate.
That's the phosphate.
Oh, okay.
That's the phosphate head. Now, we don't really talk about it when we discuss the structure, but the phosphate actually has some little, like a little carbon group here on it, but it's not really relevant. Okay, so what this would look like is circle, 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 circle. Let's say, I got lazy, all right? There's my phosphate heads. Here's my tails, right? And then I have phosphate heads and my tails. So, alright, so this is like a 2D picture here. You can see there's a middle. This is where the nucleus and everything would be at. But you could imagine if this came out and was like like a sphere, and all on the outside you had the heads, and on the inside of the sphere, all on the inside you had the heads, and then between the two layers of heads you had all the tails facing each other.
Okay. So, because of double membrane.
Yes.
Okay.
It's got 2 layers, yeah.
The inside.
Right, but the inside of the cell is mostly water too. The cytoplasm is made out of like a jelly that's sort of similar to water properties. So that's why it forms like
I
that.
was just wondering why this.
Yeah. If the inside of our cells were lipid based, then we probably wouldn't even get this second layer like that.
It would just be one.
Yeah. That wouldn't be very good for us though. Because solubility would go down for the things that we produce inside the cell. Because remember, they would repel each other?
Yeah.
All right, so here's what that looks like. We got it now. Kind of just showed you a little bit of the thing here, but do you remember discussing things that could move through the lipid bilayer and things that can't? Okay, Caitlin, what kind of things can move through the lipid bilayer without any help?
Water.
Not water.
Water.
Look at the structure. Remember, this is non-polar, and it's going to repel things like water that are polar.
So non-polar.
Non-polar things. And if you watch real closely, watch this lipid come down and go right through it. Okay? But sugars, water, salts, they're not able to diffuse through the membrane.
Hey, I heard the wolf noise.
Now, some of you might be thinking, but Miss Rutledge, the thing that we read said that water moves freely through the membrane. Yes.
That is when there is a high ratio of the volume and surface area. When there's like more volume than the surface area.
We're going to get to volume and surface area. But for movement of water, think about this. Think about this. In that reading, it said, water moves freely through the membrane. And right now I'm telling you water can't go through this membrane. So, what do I need in order to move the water through the membrane? Cameron.
Channel?
I need a protein channel. So the reading is a little bit misleading because we need something, but it doesn't require energy and the water can move freely through those channels, through the membranes. Yeah. Right. So then waste has the same problem like urea. These are impermeable to polar molecules, permeable to these molecules, and you guys already figured out the solution. We're going to have channels, right? Like tunnels. So here's a few more little facts. Cell membrane separates the cell from the environment, allows things to go in and out of the cell. Hope you feel better.
Hope you feel better.
How did you know to leave?
Okay, let's read this statement. Membrane becomes semi-permeable via protein channels. Miranda, what do you think semi-permeable means?
Maybe there's too much you can't go through.
All right, say that one more time.
Like too much water.
But just the word semi-permeable, what does that mean? Cameron?
Kind of.
Kinda.
Half as much? Like, almost half the time.
Half the time. You guys are kinda hitting it on the head. Colton?
Only under some conditions.
No, you haven't quite said it yet.
Maybe like it doesn't force on the other one.
No?
Like, I was answering. Oh.
Yeah, go ahead. I'm here.
Oh, okay. Like part, like aspects of it, or no, you want to ask part of it isn't.
Yes, let's get.
Only certain things.
Only certain things can go through.
Oh, so I was.
You guys were all kind of maybe talking about the same thing, sometimes going through and sometimes not. But that's not exactly what semi-permeable means. Semi-permeable means only some things can go through the membrane and other things cannot. So the cell is selective. It's sort of like, in the next slide I think you'll see this, it's sort of like the bouncer at a daycare.
Bounce.
Oh, yeah. They're controlling who's coming in and who's leaving, right? Or a club.
Yeah.
That's what it is.
Yeah.
Or like a fancy restaurant, you're standing at the the entrance, and you can only get in if you're so and so, right? You have to have a certain look or a certain shape, whatever. And so that's exactly how the cell is. The cell is selectively permeable, selecting who can come in and out. Now, is it always right?
Only triangles can come in.
No.
Is the cell always 100% accurate in determining what's good for the cell?
No.
No. That's how
The
the
bad stuff is in
bad stuff like who?
Like viruses.
Like a virus. Like a virus, okay? Look at this here. These colors all represent different channels. Why do you think I put them as different colors?
That's different things.
What's it?
It's different things.
Right. So the sugar here, which is a matching color, sugar goes to that channel. And what's AA stand for? Amino acids go through a different channel. The sugars and the amino acids may not go through the same channel because they have different shapes, and the channels have different shapes and sizes and things, okay? And then we've got water, we've got salts, and whites.
Wait, why did those two go all the way through?
This is the inside of the cell, and this is the outside of the cell. So releasing. We can also send proteins outside the cell, but when we're releasing a protein out of the cell, are we going to put it through a channel?
No.
Remember the little bubble system we talked about? With the vacuoles and the endoplasmic reticulum and the movement through the Golgi apparatus, and it fuses and it bubbles and pinches off? Okay, we're going to look at that in just a second because we need to talk about ways that we can get stuff out of the cell and into the cell other than through a channel. Yeah.
So if that has a specific shape, how does the cell adjust to let a virus in, we'll say?
Yeah.
So the virus will match a particular shape for a receptor on that cell, which is why.
They can transform that fast.
No, the virus. Okay, so every virus has a unique shape, and every cell receptor has unique shapes. And if the viral shape matches the cell receptor shape, or is close enough, the cell will bind it and pull it into the cell. Now, that's why, for example, that for feline, what's it called? Feline, the virus that cats get.
I think I know what you're talking about. I don't know what it's called, but I think I know.
Feline virus.
We
We
got
call it the feline virus. I forget the name of it. However, that particular virus doesn't affect people. If you have a cat that has it, you don't have to wear a mask and disinfect your house because you don't have the same receptors as the cat cells. And same thing with plants. The tobacco mosaic virus is a virus that affects tobacco plants, and people who grow crops, like farmers, if they get that virus on their crop, they can lose all their crop. But that virus isn't transferable to humans. Just because they're walking through the crop doesn't mean they're going to get sick. So the virus is specific to the receptor. Now, questions?
Why do people in Japan have to wear the masks?
When they're working around chickens,
Uh-huh.
okay, because some viruses can go from species to species. Like the flu starts out in bird intestines and can get to people. Or like the swine flu or the avian flu.
Is it going to Japan right now?
I thought maybe. All right, does anybody have questions on the channels?
Oh wait, absolutely.
Yes.
Yeah, all the things that are going through their polar, right?
Yes.
They have
If
to be
they
because
were-
if they were non-polar, they wouldn't need a channel.
Right, they wouldn't need a channel. Do you have a question about channels? Yes.
How many types of channels are there?
Lots of different types of channels, and it depends on the cell. So different types of cells, like plant cells, protists, bacteria. Then we have animal cells, and fungi cells. So there could be hundreds of different kinds, or maybe even more.
Can channels be damaged and no longer allow things through?
I suppose that's possible, but I think that
And can the channel be alternated to let different things through?
yes, and some channels. If you have a channel on a cell, you remember the cell is like a 3D ball. And so you're not just going to have one channel for a particular.
You're gonna have separate.
you're gonna have several channels all over that cell. So if one gets damaged, it may not affect the cell that much. But if you're born with a mutation, such as like the CFTR mutation, where you're not making the proper proteins, then you could end up with, you know, the whole cell not having any channels that work properly. Yes? Maybe.
The different parts of our body have different types of channels, like our blood cells.
Yes, like a neuron
Yeah.
has special pumps that pump in sodium and potassium in and out of the cell, and that's different than like a skin cell wouldn't need those pumps. Yes?
So whenever the protein is transported out of the cell, like the little like bubble thing, is that connecting to phospholipids or columns?
Phospholipids. And I have a picture to show that in a few slides. All right, so let's keep going. You've seen these amino acids before.
Yes, we have.
Mira, what do you know about these amino acids by looking at their R group?
Or R group?
Mm-hmm. The side chain.
Oh.
This, huh? This. Here's a clue. Do you see the clue? This is like Blue's Clues left right here.
Just make it blue. Just put a clue. Put the
Oh, they're not. They're all non-polar
Yeah, just figure that out.
because their side chains are mostly made out of
Carbon and
carbon and hydrogen, hydrocarbons, right? And so, back when I taught proteins, I told you guys there's polar amino acids and there's non-polar amino acids, and I showed you this slide, and then I showed you this slide, and we saw that this polar amino acids have oxygen and a lot of nitrogens, which create uneven sharing of electrons, which causes them to mix nicely with water. And that was pretty much what I left it at. And you guys weren't really curious about maybe the uses of polar and non-polar amino acids, but today it matters. So, look at the cell membrane. Think about this. If I took amino acids, and this is a protein, I'm just showing you the ribbon structure now, okay? So instead of a blue like oval here, this is the ribbon structure, but it's still a protein. These amino acids right here, they're in contact with what? The fatty acid tails, which are non-polar. If I tried to put polar amino acids next to the fatty acid tails, what would happen? They're going to repel each other, and this protein would like pop out or pop into the cell. It would be like squeezing it and, you know, flip out because they'd be repelling each other. But because we have different classes of amino acids, I can build a protein and put non-polar amino acids through the middle area where it's in contact with the tails, and I can put polar amino acids at the top and at the bottom where it would come in contact with things like water or sugar, so that when I'm ready to move things through the middle, and the middle is kind of a hollow space, sort of like could be like a tunnel, or it could be filled up, and when something comes in contact with you, the bonding would change the shape of that protein. Something would bond here, and the shape would change and allow it to go through. It's fancy, huh? So that's what that looks like. Non-polar amino acids in the middle, polar amino acids on both sides, which gives everybody a happy place. The changing shape of the protein when something binds to it is called the conformational change. Probably not going to be on your test, but I figured I'd throw that out there. All right. At the beginning, I showed you this picture, and somebody asked me, but what are all these other molecules? What are these things in blue that have a little hole right here? Those are proteins, right? Proteins. Is this a protein? Yes. Okay, good. I'm going to show you some labels. These green things here are cytoskeleton. Cytoskeleton gives the cell its shape, kind of like bones give us our shape. If we didn't have any bones, we'd be like little flabby sacks. Probably like a pile of stuff on the floor, right? So bones give us our shape, much like cytoskeleton gives a cell its shape. It's not made out of the same thing bones are made out of. Cytoskeleton is actually made out of protein, different kinds of proteins. Yeah.
On like the drawings of the cells, like on that thing, they had like these little tiny X things. I don't know how they were like little sticks.
Like a little
that were,
T?
No, or maybe I have it. Like I didn't know what they were, and I didn't know what organelle they were.
Was it an animal cell? Like a rounder cell? Or
No, it was like here. Well, I may not have it, but it was like on the
there?
no, it was on here. I was those things.
Yeah, that's cytoskeleton.
That's
cytoskeleton. Yeah.
Okay, that's what I thought.
But if they actually drew all of the cytoskeleton on the picture,
Then
you wouldn't be able to see anything else because it's like a whole framework. Yeah.
I thought that like purple spirally thing and purple straight thing, I thought they were both the same channel. Like no,
This,
that,
this,
no,
that?
yeah, those two things. I thought they were both channels.
Yes, but their structures are different.
Oh, because
so they're
Yeah.
all channels, but they're just different.
Different types because they probably transport different things. All right. These are transmembrane proteins. These yellow things are cholesterol, and I heard you guys arguing a few minutes ago about what cholesterol does. Frankie, tell me how cholesterol, what is the function of it in the cell membrane? Colton, can you help her?
Doesn't cholesterol help to hold it together and keep it from being spread out too much, sort of like ropes
Right, so
holding it
when
down?
you can imagine when molecules speed up, temperature is a measure of how fast molecules are moving, right? So when you heat up a cell membrane, those molecules have more energy and they tend to want to spread out more because they're bouncing off of each other. And so a cell membrane, if you heat up too much, would spread out a lot. But the cholesterol here that's attached to the tails actually holds it together when it gets too hot. Now, when it gets too cold, molecules slow down and they condense, except water. So when they slow down and get closer together, the cholesterol holds them apart so that the cell membrane doesn't become too hard or too rigid. So we say that the cholesterol helps maintain the fluidity of the cell membrane, the integrity of it. Okay, now we're going to switch over from structure and we're going to talk about diffusion. What is diffusion? Mira?
Fusion is like, I don't, it's like moving to the something else, like spreading. Yeah.
Okay, the spreading out of molecules, right? Diffusion is when they move from over here, what do I have over here, and then compare that to over here. So you've got on the first picture a lot of molecules on one side, and then what happened? Haley?
They transferred to the.
Right, so that they were more evenly distributed, right? So basically, diffusion is the movement of molecules from high concentration to low concentration. Does diffusion require energy?
No.
No, we don't have to put in energy to make this happen. If I fart up here, sorry guys, my fart molecules are going to be bouncing off of each other, and that energy of them bouncing off each other, just the natural energy, is going to cause them to spread out more. Okay? And they're going to keep spreading out until the room has evenly distributed fart molecules. Okay?
We got this.
Now,
Can I demonstrate that?
no. Did it take any energy for those molecules to move around? Did I have to blow them or swipe them or something? They just did it because molecules are constantly moving and they're bouncing off of each other and they're happy when it's equal. Now, if I put a hole in the wall, to Miss Schroeder, if I punched a hole in the wall, okay? Well, first let's say there's no hole. Can my fart molecules move through the wall?
No.
All right, but if I put a hole in the wall,
Yep. Yes.
they could move through the wall. Would it require any energy to move them through the wall? No. Eventually they would just sort of diffuse through the wall, right? So we have simple diffusion, which is the movement from high concentration to low concentration. Down here in the bottom left corner, that's my fart, and there it is spreading out in the room. Okay. Spreads out evenly. It's called passive transport because there's no energy required. Simple diffusion. I'm going to come back to the movement of water in a second.
So will it be equal in each place then? Or is it try to, like, it does its best to be as equal as it can everywhere?
Yes, equal as it can everywhere. And there may not be enough molecules to completely even out everywhere in the room because maybe they will stop bouncing off each other and start bouncing off other molecules in the room. And so
There was
So you were breathing in molecules of throat smell. Smell. The molecules that cause the odor. Alright. Facilitated diffusion is what I described with the hole in the wall. Molecules can move, but they need a tunnel to move through. Does that make sense? Molecules can go from my room to Ms. Shorter's room if I put a hole in the wall. That's facilitated diffusion. I helped the diffusion of fart molecules into her room.
But what if it was already there?
If the hole was already there, it would still be facilitated diffusion.
Okay.
Because it was necessary for it to.
Yeah, that hole is required for molecules to move through it. Now, we don't have to do any work. We don't have to,
Yeah, you don't have to like break anything to like get it to you, but there has to be something.
Right, so in a cell, water or salts or things that are moving to equal a concentration that are, if it's a polar molecule,
They move in there without any energy though.
Right. Right, they move through the channels. Now, I said I was going to talk about water in a second. Let me go back to this picture. Okay, first of all, facilitated diffusion. We need a channel to help with the movement, but there's no energy required. Let me go back to the previous page. Look at this picture, diffusion and osmosis. A lot of times kids get these terms mixed up. If I took red food coloring, and I had a beaker of water, and I dropped some food coloring in it, the food coloring molecules that would spread out, right, and eventually the whole water would be red, correct? Is that diffusion or osmosis?
Diffusion.
Diffusion. How do I get osmosis to happen?
What is osmosis?
Osmosis is the movement of water across a membrane to equal the concentration.
What would they need a channel to get any kind of energy?
It doesn't have to require energy.
What was the question?
All right, so osmosis is the movement. Okay, look at these green dots. Let's say these green dots represented salt. And we want the salt concentration to be equal on both sides. But let's say that there's no salt channels here. No salt channels, but we can move the water. If we can move water, which way would we move the water to equal the salt concentrations out? Caitlin?
Move it to the right.
I will move it from over here to over there, from the left to the right. Think about this, guys. If the only thing you can move is water, and you want to make these sides more equal,
Oh, you make it so that.
right? You add water to from the left to the right, it's going to dilute this side. And if you're taking water away from this side, what's going to happen? It gets more concentrated, and that's going to equal out the solutions. So water moves across a membrane during osmosis. Yes?
But wouldn't that require energy to force something to go into a higher concentration?
But we're not moving the salt.
But there's a higher concentration of water.
None of water. Over here, there's a lower concentration of water because there's more salt.
So is that when they have like one of those water channels?
Yes. Yes. The water channels.
Yeah, so that's only when it has a water channel?
So there, yes, that's when their water can move. Now, in your cells, it's much easier to move water through channels than it is to move other things. So usually what will happen with the cell is if we put it in something that's got solutes, the water will move in and out of the cell. And you see this when you're in the swimming pool and your fingers get wrinkly as a result of your cells being placed in a different solution. Okay? We're going to talk about that. So osmosis is the movement of water across a membrane to equalize a concentration. Does not require energy. It does not require ATP or anything. It happens naturally because things want to be equal.
So it's water with the concentration to what?
Equal concentration. So, simple diffusion, facilitated diffusion, and osmosis are all passive things. No energy required. But, what if I want to move things against the concentration gradient where I already have a high concentration, like you were thinking of? Let's say I have a lot of salt over here, and I want to add more salt where I already have a lot of salt. I'm going to have to put in energy to do that, and that's called active transport. And this picture is upside down on this slide, so if you can imagine the purple things going the other direction. I think I need to rotate the picture. So active transport is moving against the concentration gradient. And so you guys see right here? This shows the molecules moving this way, but that's not right for active transport. So to move active transport, these little, where there's not much concentration, these molecules would be moving this way towards the more concentrated area. We see this in neurons. Neurons live in a sea of ions. And there's potassium and sodium ions. And whenever you have a stimulus, like you feel something, those ions go in and out and settle really fast. And then to restore it, we have to pump the ions back in and out of the cell. And then you can feel it again. This happens really quickly. Which is why you can only feel so much stimulus in a particular time. It's kind of like dominoes. When you have a stimulus, it's like knocking over the dominoes, and then you can't keep knocking the dominoes over. You have to set them back up first before you can knock them over again. And the setting back up is referring to pumping the ions that requires energy in or out of the cell. Cameron?
So could you just get tired by just feeling too much? Or like, is that relatively low amount of energy it takes to?
It's a smaller amount of energy compared to like how much energy your muscle uses. Question?
So caffeine, does it make your body think that it has energy to be active, or does it supply it with temporary energy?
Caffeine does not supply your body with temporary
So it
energy.
makes it think that it has more energy, or therefore making you...
I think caffeine has to do with something in the brain and not really like your body and energy. Question?
Marie said that.
That is caused by the release of special hormones and chemicals in your body that block pain receptors.
Because it hurts.
Why do we have
The
that all
same.
the time?
So if you break your leg and you fell off a cliff and you're about to get chased by a bear, and you get up and you run with a broken leg, the adrenaline and the endorphins in your body block the pain sensors
So you can get away.
so that you can get away. But the message that there's pain is still being sent. Your brain is just not receiving it right now.
You'll just get it later.
You'll feel it
Definitely
later.
get it later.
If you're an athlete, you probably know this. You can be, if you're really in a high stakes game or something, maybe you guys are too young to know, but you can get an injury, or you can really like play through a lot of pain
Yeah.
because you have so much adrenaline, and then like within 30 minutes after the game is over,
Yeah,
you
like
really start
when you're playing.
Yeah, I just
That exact thing happened last week. I was playing a game.
I really
and
didn't
it was
play
the second set, and it was like I played for an hour and a half, and then I played another hour and a half. But as soon as I got out of the court, then I felt like, oh my god, I
Stop
found out I
doing-
pulled a muscle.
yeah.
All
right.
Why did you
So let's recap. This is sort of a review slide. If I'm going through this and you're like, okay, then you're pretty good. But if I'm going through this and you're still not sure, then you might need to go back and relook at this to make sure you got it. Passive transport has two kinds: simple diffusion and facilitated diffusion. Simple diffusion is just the movement of molecules from high concentration to low concentration. What is different about facilitated diffusion, Josh?
You actually have a tunnel to help
Right.
it
We need a protein channel to help the molecule move through. Still requires no energy. Active transport, Karen, what's different about active transport from both types of passive transport? It's harder to get through, so we need, Caitlin?
Energy.
We need energy to make it happen. Are we moving with the concentration gradient or against it?
Against.
Against it. Good. Going to use a pump for that. Requires ATP against the concentration gradient. Now, none of these things mention osmosis. These are all diffusion. Osmosis is the movement of water across a membrane. And here we're not really talking about moving water across a membrane. So, review. Can you go back?
Like the last line.
This one? Simple diffusion, no channel needed. Facilitated diffusion, channel needed, moving from high concentration to low concentration. And then active transport is when we have low concentration and we're going to use energy to pump it to a high concentration.
So like, for a simple diffusion, it would basically, in terms of the cell membrane, that would be lipids, and for a facilitated, it could be like salts.
Right.
Okay.
Cameron?
So is osmosis just a type of active transport?
Osmosis is not active transport, does not require energy.
So is it
Oh, yeah, yeah.
It's its own type of transport.
Is it even considered a form of diffusion?
It's the what?
Is it even considered a form of
No,
diffusion?
no. It's separate from diffusion. So, moving large molecules, I'm just going to take like 30 seconds on this slide because we've already kind of seen this. Imagine back to the Golgi apparatus, fusing with the cell membrane. This would be a protein. This would be a little bubble that formed, okay? This picture can be seen both ways. The way if you think about the red things moving out of the cell, it's called exocytosis. If you think about the red things moving into the cell, it's called endocytosis. So the cell can actually wrap part of its cell membrane around a molecule to engulf it into the cell. Question, Miranda? If they're not leaving through a protein channel or simple diffusion. You see the difference? These molecules are usually too big to go through a protein channel. Like if you make the chains for a hemoglobin, that's a big protein. You're not going to have a channel big enough to fit that molecule through it. So we're going to release that through exocytosis to let out a big molecule. Yeah, that could be an example of this, but through endocytosis we can take in large molecules as well. In fact, white blood cells that engulf pathogens, a white blood cell might come up and just kind of wrap around a pathogen and engulf it. Marcella?
No, that's
Creates a bubble.
The bubble, okay.
And then once inside the cell, different things can happen with this bubble.
Okay.
Sometimes digestive enzymes will be inserted and this will be dissolved in the case of like white blood cell. Or these might be necessary proteins that will be sent to a different part of the cell to be used somehow. Questions?
What is this?
Secretory is whatever you secrete. Another example, this is a good one. Pancreas. Your pancreas has beta cells that secrete insulin. And so insulin would be a protein that you would secrete from a cell. Question?
So if it was going into the cell, it would be the bubble would be made out of phospholipids because it's
That is
part of
correct.
membrane? If it was
All
the
of
other
these
way,
membranes inside the cell are